9.
The Radio Wammo Breakfast.
Live from the New Scientist Labs, Janine Young, newscientist.com.
Yes, from Melbourne City, Janine Young's back with us with the latest in new science news. Morning, Janine.
Good morning to you.
And good morning. And a big science story over the past week or so, of course, has been this this outbreak of, I want to say salmonella. Is it salmonella?
No, it's Escherichia coli.
E. coli. So it's more to do with poos, really, isn't it? Actually, is it? Because we're often told that E. coli, say E. coli in water, is feces. Is not always the case?
E. coli is a fecal bacterium.
Yeah.
You do find it all the way through your guts. We're talking about gut bacteria a lot this year when you think about it.
Yeah.
So we should all be, fairly good with this sort of thing.
Yeah.
Yeah, it's found in gut bacteria. It's also found in fecal bacteria. But if it's going to be in your gut, then of course it's going to pop out the other end as well. E. coli will often live on your gut wall. So, it can be right across the entire digestive system. And as we've heard before, they're actually really important for being able to digest food, and they're actually on the whole generally good for us. Until you get a strain like the one that's popped up in the last few weeks. It's killed 22 people in Germany and made more than 2,000 sick. This one has picked up something that's called Shiga toxin. It causes really nasty, nasty effects when it actually transfers off the gut wall and this toxin goes into the bloodstream. It causes hemolytic uremic syndrome, which is kidney damage. People have known about these Shiga toxin E. coli for a while, and frankly, they're nasty bastards. I don't mean to get too callous about this, but they really are. They're they resist 8 different types of antibiotics, although it's kind of a moot point anyway, because the antibiotics that people try to put on them, instead of just going, oh my goodness, I'm going to curl up and die, they actually have an SOS reaction and they produce more toxin. Now, so it actually just makes the situation worse.
Is this a result of antibiotics use over the decades?
Certainly antibiotic resistance in a number of different other strains, absolutely, yes. This one, it's just picked it up. And that's the thing about bacteria, they swap around genes all the time. We've talked about the flu epidemic that went around a couple of years ago now. They reassort and they mess their genes around all the time. Bacteria do the same thing. They're able to shuffle their genes quite regularly and quite easily. This nasty strain hasn't been seen before, and a lot of the techniques that are out there to be able to pick these sorts of things up wouldn't have found it anyway. So it's a bit of a nasty situation because the people that are affected by it are in a catch-22 because the antibiotics don't work. As I was about to say, there is one class at the moment, the carbapenems, and I must admit I'm not big on my pharmacology anymore, but they're cautiously using that one category of antibiotic. They seem to be having some success with it. But they're very cautious because, as I said, they shuffle their genes around. They pick up resistance all the time.
Yeah.
Now, and I have a list, a scary list actually here, that Staphylococcus, which is a common bacterium that you find, it causes throat infections and that sort of thing. It's a common bacterium. But nearly half of the supermarket meat in the US has been found to have nasty strains of Staphylococcus on them. Vancomycin-resistant Enterococcus is rampant in antibiotic-treated chickens, apparently. MRSA, most people would know that this is a nasty one in hospitals. This is another Staphylococcus. There's now a strain of that that's able to evade DNA testing, so it means that they can't find it in the first place.
The ultimate criminal.
Well, yes. It has no fingerprints. There's some nasty stuff. Tuberculosis, I think a lot of people would know about that as a problem that's been increasing for the last little while. While the cases are going down, antibiotic resistance is going up alarmingly. 25,000 cases of an XDR strain, a particular one that's it's resistant to almost all drugs. Now, tuberculosis hasn't been a serious disease for a very long time, but apparently it's so bad in London now that they're actually reconsidering whether or not they should be vaccinating children against tuberculosis. This is a serious, serious problem because antibiotics were the wonder drug of the 1950s, and research into them has really slowed down. In some cases, it's practically ground to a halt.
So is it?
You don't need any more.
Well, is it time to start looking past antibiotics? Are there any other solutions?
There are, and that's what this article is fundamentally talking about, aside from scaring the pants off people about how bad this is. There are other ways that we can tackle these sorts of diseases that are caused by nasty bacteria that are allergic. I keep saying allergic. That's not right. Resistant to antibiotics. Being able to block the toxins that bacteria produce. Just out of interest, things like food poisoning, it's actually not the bacterium itself that causes the problem. It's actually the toxin that the salmonella produces that's actually toxic. That's the problem. That's why you get sick. So if you have the ability to block the effects of these toxins, if you can stop it, for example, getting from the gut wall into your bloodstream, then you're going to have fewer problems than if you can't, for example.
Yeah.
Vaccines against the toxins is another possibility. Vaccinating cattle against these sorts of toxins. Being able to take bacteria off food. There are particular types of viruses that munch down on only bacteria. They're called bacteriophage or bacteriophage, depending on where you're from. I learnt phage in university, so I'll stick with that. Yes, we're sounding proper today. They eat nothing but bacteria. And so there are some treatments available, particularly in the US, although their efficacy isn't entirely proven, that claim to be able to remove bacteria from the surface of things like meat. They can then replace them with other bacteria that aren't nasty. So that's an approach as well. But we do need more research into new antibiotics and looking at different mechanisms instead of the ones they're using now, because antibiotic resistance is a massive problem. And the fact that we are, and don't get me started on this for too long because I've been jumping up and down about this for years, using all these antibacterial sprays on our bench tops and on our hands and all that.
All the scary bugs around the toilet rim, all that kind of rubbish.
Well, in some student houses, yes, I would agree. It is pretty nasty stuff.
Yeah.
I've lived in a couple of them, but that was a long time ago. Keeping clean is important, but that's hygiene as opposed to being fanatical and using antibacterial everything everywhere.
Yeah.
There is such a thing as too clean, and it does put selection pressure on bacteria to resist these bugs. I mean, is it surprising that they're doing what comes naturally, if you want to put it that way?
Trying to survive.
If you put a pressure on them, that's what they're going to do.
Yeah, exactly. Now, before we go, just a quick look at this article about genetic breeding, because often we think of genetic breeding as a sort of a modern thing, this idea that we can create crops that will yield more or be resistant to pesticides, that kind of stuff. But it's actually been happening for, 10,000 years, Janine?
It has, yes. That's how a lot of our plants have been domesticated, how we've ended up with the wheat and the rice and the other kind of crops that we have at the moment. It started a long time ago. Research out of Kobe University in Japan is showing that there's a particular type of sticky rice, Oryza sativa japonica, just to make sure that we're right, was being selected for higher yields. And what these farmers unwittingly did was selected for just one gene. It was an SD1.
Yeah. And
they now have the evidence in their sticky rice that, 10,000 years later, that there is less variation around this one gene than there was in all the other clusters that they've looked at across the rest of the genome. So they didn't realise that they were doing it. But yes, they were selecting for particular traits as part of the domestication process, because long grain rice and sticky rice were domesticated about 10,000 years ago in China. And who knows, they've actually done something they didn't even realise. Selective breeding these days generally takes 20 or so years if you're intensively trying to hone in on one particular trait. But what these farmers were doing over time was going, well, that one grows really well, that one is fairly hardy and produces a good yield, we'll keep growing that. That's all they did. And instinctively what they did was just pulled out one gene. Makes a lot of sense.
I mean, if you're growing some beans at home, you're always going to select the seeds for next year that crop the most. So, it makes a lot of sense.
I've certainly done that with my tomatoes this year.
Yeah. Janine Young, you'll find those stories in New Scientist magazine coming out next week. And we'll also talk to you next week. Thanks.
See you later.
Here's